Okay, thank you very much, Paul. So, I come from the Sheffield University, where we have an active touch laboratory, and also the Sheffield Centre for Robotics, which we founded last year. And I want to tell you about the work we've been doing really in the last 10 years. My training originally as a psychologist before I moved into artificial intelligence and computational neuroscience. And what I do now is to try and merge the study of biological systems with the development of synthetic systems that mimic the biological systems in order to help us understand them better. So the talk will have these four sections to it, and they are really, the first one is an introduction. So people that have been to BCBT before will know the work we've been doing on active vibrissal sensing in mammals and how we've been trying to translate the principles and ideas from that into whiskered robots. And these robots are what we might call neurorobots. They're controlled by models, often quite high-level models of systems that we find in the mammalian brain. And then, since there's been a focus on evolution this week, and I'm myself very interested in evolution, I'm going to talk a little bit about the evolution of this system and how it arose, I would suggest, relatively early in the evolution of mammals, and therefore could have been quite influential in the evolution of the mammalian brain. And finally, I want to talk about the most recent work, which is about how we might control our spatial attention and the insights we might get by studying this vibrissal system of the rat and the synthetic models of robot models of the rat vibrissal system to understand something more general about the control of spatial attention in mammals, including ourselves. So before I get into the research, I want to just acknowledge all the people who have collaborated, and particularly Ben Mitchinson, who is the co-author of nearly all the work that is done here, and Martin Pearson, who has built most of the robots you'll describe. Charlie Pearson actually built one of them. Various other people, Paul Dean and John Porrell, did work on the cerebellum, Kevin Gurney on the basal ganglia, Stuart Wilson built cortical models, Kendra has done a lot of the behavioral work, and Robin has been leading the behavioral work in our group for the last 4 or 5 years. And these guys from Bristol Robotics Lab build all the robots, and we collaborate with them to develop the control algorithms for the robots. So part one, active vibrissal sensing as a window into attentional processing. So I'm going to tell you something about the vibrissal system, and for those of you that haven't seen it before, show you what the rat is able to do using its vibrissae. So before we get into rats, I just want to talk a little bit about human attention. And of course, when we study human attention, we usually study the eye movement system. And this is classic work from Albert Yarbus, a Russian psychologist. He was doing this work in the 50s and the 60s, and he wrote this very well-known book, Eye Movements and Vision, in 1967. And this was one of the examples that he used to study spatial attention in people. So if you present this picture to a human subject, it's called the unexpected visitor, then and you measure where they move their eyes, so you measure the saccadic eye movements that position the fovea of the eye at particular points in the image, then this is a typical pattern of eye movements that you would get. So this is obviously different movements all strung together, but you can see that the focus is on the key players in the scene. The woman standing up to welcome the unexpected visitor here, onlooker, various other points in the scene.
There's not a fixation at the feet of the unexpected visitor.
I guess there is. Maybe this is because the implication of movement. So maybe you're trying to infer some direction from here by looking at the feet. And this is actually a quote from Albert Yarbus, what he wrote in his book. Records of eye movement show that the observer's attention is usually only held by certain elements of the picture. And clearly, they're the psychologically important elements of the picture, not necessarily the most salient or the brightest points in the picture. And he said, eye movements reflect the human thought processes, so the observer's thought may be followed to some extent by records of eye movement, the thought accompanying the examination of the particular object. It's easy to determine from these records which elements attract the observer's eye and consequently his thought in what order and how often. So there's a really strong claim here from Albert Yarbus about the interpretation of these saccadic eye movements, where the eyes are positioned, and that this tells us something really useful about what people are thinking. What the observer is thinking when they're looking here is indicated by where they're fixating. So, and of course, if there are lots of salient locations to look at, here's another famous picture from Yarbus, then they're visited sequentially. So you would go from looking at the eyes to the mouth, maybe back to the nose and back to the eyes again. And there's a combination here of overt and covert attention. So there will be some attention which is not revealed by the eye movements, because you can inspect the different aspects of the picture without moving your eyes or without moving them very far. And there's also a combination of exogenous and endogenous influences. So exogenous influences would be things like bright spots or contrast points within the image, which might drive your attention to those points. And endogenous influences would be things like your motivation at the time. So referring back to the previous talk, if you thought this was an attractive picture of a woman, you might gaze at her for longer. So I'm going to talk about a different system. It's still a mammalian system, it's still a sensory system, but it's a different system in which I think we can also study attention. And although we've studied this system a lot in neurobiology, we haven't studied it necessarily from this perspective. So I'm going to talk about the rat sensory system. This is Ratatouille, a rat as imagined by Walt Disney, gazing out over Paris. But in fact, when Ratatouille gazes out over Paris, the scene he would see would actually be much more like this. Because his visual system, as the previous speaker mentioned, is not as good as ours. The resolution of it is extremely poor, and indeed he won't even see much in color. He has very few cones, so his view of Paris will be much less appealing than ours. He will see it this way. So rather than relying on vision, which in rats and other rodents is often used as a defense system, it's for detecting perhaps movement overhead that might be threatening, the rat is using its vibrissal system much of the time to explore the world. So this is a rat exploring on a windowsill, actually in my house. It's a pet rat. And you can see that the whiskers are moving all the time that he's moving. The whiskers are moving back and forth very quickly. This is real time. He's reaching over the edge of the windowsill with his whiskers. He's exploring this wooden cup. So whenever he's moving, the whiskers are an important aspect of how he investigates and explores the scene. And this scene is filmed in daylight, but we film many times in infrared or with animals that are genetically blind, and we see very similar whisker motion. So there are interesting parallels to how humans use touch, with our fingertips. I won't explore those today. But also, a useful thing about the system, a good reason why we should study it, is that the pathways within the brain that control the movement of the whiskers and that relay vibrissal information to the higher levels of the brain are very well studied.
Can I ask a quick question?
Yeah.
Are the whisker movements exaggerated in the genetically blind mice?
No, I would say they're very similar. You might perhaps see more exploration in the genetically blind mouse. We haven't quantified it. But it's certainly from observing the videos, you wouldn't know what was blind and what was sighted. So the argument is that this is a good model system in which to try and understand the general principles of sensing in mammals, active sensing, active movement of the sensory apparatus, and also, I will say today, to understand attentional processing in mammals. So we've done a lot of work to try and study and quantify the active sensing movements that these animals make. And so we generally use a high-speed camera. About 500 frames per second is good. We have a purpose-built light box, so we can flood this area with light. We have a mirror at the back, so we can see a back view as well as a top-down view. We use these sighted animals, but we also use genetically blind rats. They're a strain called dystrophic Royal College of Surgeons rats. And what we've often done is just to put the animal in this box and film opportunistically. We wait till he comes under the camera and film him then. And sometimes we put objects in the arena to see what happens when the animal interacts with the objects. So the rat has two qualitatively different kinds of whiskers. Let's just go back to this picture. No, sorry. I haven't got it. Let's just go on to this video. So you see these are the macrovibrissi, the long whiskers, and he has about 30 on each side of the head. And in each of these videos, you see an animal that's been trained to run up a corridor, and most of the time, there's nothing in this corridor. So he just runs up the corridor, and as I'll show you later, he does a lap, he gets some food reward, and then he runs up the corridor again. To make these videos, we took this trained rat and we put an object in the corridor that he wasn't expecting. When he ran up the corridor and contacted it with his long whiskers, so you'll see a contact here, then he immediately slows down and turns and orients and explores the object with his whiskers. So this is an animal which is genetically blind. So he's not using vision to explore. We know that he's used touch on the vibrissi to encounter and recognize that this is a novel object, and then he's orienting to explore.
Is he being rewarded there, or?
He's not being rewarded for exploring. He's just being rewarded for running in a loop. You can see in this one we do the same, but we've just put a step on the floor. So the step is unexpected. So he's running along. He encounters a step with the whiskers that are trailing on the floor. You'll see that in a later video. And then he orients to explore that object with his whiskers. So you can see he slows down very quickly and starts to explore. What he's doing after he's oriented is to position these very short whiskers on the chin and on the lips, which are called the microvibrissi, and they're much more densely packed than the macrovibrissi. He's positioning them so that he can explore an object with this, if you like, foveal region of the vibrissal sense. So here you can see what we like to call dabbing of the microvibrissi onto this plastic puck. And what you can notice also is that when he dabs with his microvibrissi, the macrovibrissi come down at the same time. So, and they make contact with nearby surfaces at the same time as the microvibrissi are contacting. So what he's really doing is getting a series of discrete contacts. This is slowed down, but he's doing this about 8 to 10 times a second. He's dabbing with his short whiskers, and the longer whiskers are encountering the surrounding area. So you have a high resolution area in the macrovibrissi where you're getting lots of samples, and as you go out, you're getting lower resolution in your sampling in a nicely graded way.
The short whiskers also go to the barrel cortex?
Yes, they do. And there are barrels also for the short whiskers. So what I want to try and persuade you of, the control of the long whiskers, the macrovibrissi, tells us quite a lot about the spatial attention of the animal. So when the animal turns and orients, we're already getting information about his attention. His attention has been grabbed by an unexpected object, and he turns and orients and explores it with his shorter whiskers, as well as, of course, with the other sensory receptors around the mouth, and he sniffs with his olfactory system and so on. But if you watch this animal here interacting with this perspex block, then you see that he's also changing the way he moves his long whiskers in order to help use those long whiskers better explore this more complex shape. So if you watch again, you'll see the whiskers on this side moving a long way to try and contact the perspex block. If you wait while he comes around again, you'll see that the whiskers on this side start to move a lot less when he's contacting the block, while those ones reach round. So from this, we can infer something about what's interesting in the world. The animal's interested in these surfaces that he's encountered, and he's trying to generate more contacts with them.
Does this have anything to do with theta waves in the hippocampus?
Probably not, certainly. David Kleinfeld's looked for a correlation and hasn't found one. But I don't think that's completely confirmed. Matthew Diamond has said that there might be a correlation under the right circumstances. So in this video, it's a similar, now encountering a curved object, and you can see again the whiskers being moved so as to make more contacts with the object, and I'll provide more evidence of that in a minute. Also, what I'd like you to notice is that when he explores over the top of this object, he's able to move the more rostral whiskers somewhat independently from the more caudal ones. So you'll see here the whiskers reaching across the top and these ones at the back being held back. So we don't see this very often. Most of the time, the whiskers on each side appear to move together, and most of the time, the whiskers on the two sides appear to move synchronously, although the synchrony can easily be broken. But actually, for each whisker, there's a muscle that pulls it forward. And so there's many degrees of freedom for positioning the whiskers. So people maybe 20 years or 30 years ago thought that there was just some stereotype whisking motion. That's definitely not true. There's very precise, I think, richly varied, I think we can show it's goal-driven and it's feedback-modulated behavior. I want to argue that this reflects internal attentional processes, and it's driven both by these exogenous inputs, so the shapes in the environment, and endogenous influences, as motivation and so on. So I think that it's comparable then with the human visual system as a potential model of mammalian attention. So I just want to dissect some of the components of what's going on when the animal's controlling its macrovibrissal. So this is a result which was discovered by Mitra Hartmann and Blythe Towell, but this is actually replicated here in our lab. This is a 16-day-old rat pup, and he's circling. You can't see the whiskers here too well, but this is actually an adult animal. But what happens when the animal is circling is that there's asymmetry in the whiskers. So normally the whiskers would be symmetric with the position of the head, but when he's circling, the whiskers on the side away from the turn are pushed further round in the direction that he's turning, and the whiskers in the direction that he's turning in are pushed back. So if you like, when he's turning, rather than having whiskers out like this, he has his whiskers like this, so that he's moving his whiskers to anticipate possible contacts in the direction that he's turning in. So I showed you already some evidence that he can vary the velocity of the whiskers on each side, and that they don't all have to move together at the same speed. And we have other evidence showing that what happens when he encounters an object is that the whiskers at the front move less and the whiskers at the back move more, so that these whiskers, when you view them, appear to come further up. So we can describe it as a reduction in the whisker spread. If you take the angle or the angle between the frontmost whisker and the backmost whisker, then that angle is reduced when he's exploring an object. And it's being reduced because the velocities of the whiskers are changing, so the back ones are moving faster relative to the front ones. So another finding that you've already seen illustrated in one of those early videos is the effect that asymmetric contacts with objects has on the movement of the whiskers. So this is now a wild rat. So he has actually bushier whiskers, I think, because he's wild. You'll see nice symmetric movements. But as he explores this object in an asymmetric way, you'll see that the whiskers on one side, so he's going this way, the whiskers on this side begin to protract a lot more, and the ones on this side a lot less. So you can see these ones pushing round to try and find the objects, these ones moving a lot less and lightly touching.
So is the nose also, the tip of the nose also being used as part of the sort of tactile perception mechanism? It seems like it doesn't hesitate to slow down when it's banging its nose against the object.
Well, the nose is close to the object. Certainly he's sniffing the object. So that's definitely part of what he wants to do, is to, and the sniffing is synchronized with the whisking. So I would say he's not pushing the tip of the nose against the object. I think he's getting just very close to it and sniffing it. But certainly the whiskers, the microvibrissal are just below the nose, and they're contacting. So this video is really showing the same thing, that the symmetric whisking is replaced by this asymmetric whisking. So on the side that's closest, less movement. On the side that's further away, these big whisker sweeps as he's trying to find the other edge of this object that he's exploring. So this is a very slowed down video of the first contact with an object. So what I want you to notice here, and this will be repeated several times, is that initially the whiskers are synchronized on both sides. Then there's a contact here on this side, and then the whiskers on the side that made the contact, the ipsilateral side, start to retract almost immediately after the contact. Actually, it's about a lag of around 12 to 14 milliseconds. And the whiskers on the contralateral side don't contract at that same time. They contract was what was probably due to be the end of the whisking cycle. So you can see this is the ipsilateral side, and this is over 20 or so different videos. There's always a retraction around 10 to 15 milliseconds after the contact. On the contralateral side, there's no relationship between when the whiskers retract and the moment of the contact, which is 0 here. So this suggests to us that what the animal is doing is it's making contact, but it doesn't want to make hard, strong contacts. It doesn't want to bend the whiskers hard against the surface. It wants to make a light touch. Certainly in this experimental paradigm, he's just exploring. A light touch gives him good information. He can pull the whiskers away and sample again in the next whisk cycle.
And when he samples again, does he whisk out to a smaller angle?
When he samples again, he seems to control the whiskers to make a light touch, and we'll come back to that.
He knows where it will be, and it can compensate for its body motion and things like that.
That's our hypothesis, yeah.
And you think he's already got some impression of the surface orientation from that first contact?
That's certainly our hypothesis. We'll show you, we have, the model seems to support that. I'm not sure if we have direct evidence from the animal. We have some ideas about where you might get it. So this is an overview of the apparatus I showed you before. So that in the experiment I showed you before, he was running up this alley and contacting an unexpected object here. And this is the full apparatus. So there's a door here that's operated by a light beam, another door here. When he comes down here and breaks the light beam, the door opens. He comes up the alley. Another light beam is there that opens this door. And then he comes down this corridor and he gets a pellet. So in order to get a pellet, he has to complete a circuit. And rats that have been food deprived for a few hours will work for maybe about 40 laps to get pellets before they're satiated. So during that time, we measure their whisker movements as they run up the corridor here. So if we put a naive rat into this apparatus, he's never seen it before, then, and now we've got an overhead view. Sorry. We have the overhead view here of the rat, and we have a side-on view in a mirror, in an angled mirror. And so you can see he's moving slowly up the corridor. He has his nose very close to the ground. He has a quite crouched position. He's exploring very carefully. If you look at his whiskers, he makes relatively large sweeps of the whiskers, which are fairly typical of the exploratory whisking that we see when we put an animal in an environment where there's novel objects or where he's just exploring and is not in any hurry. So this is an animal that's had 2 days in this apparatus, and now he's less interested in the corridor. You can see he's moving faster. His head is lifted up quite a lot. He's making longer strides. And the whiskers are still moving, but not so much as they were before.
Do they tend to do wall following, following one wall or the other, by maintaining tactile contact with one wall? Or?
Not especially. I mean, they don't stay close to the walls, certainly.
How do they know they're in the middle of the corridor? If they're blind?
I guess they remember the corridor, but...
So the first time around they'd be bumping?
The first time around they'd be exploring all over the corridor, finding out where the walls were. So this is after a week in this environment, and now you can see that this guy is really bombing up the main alley here. He's making these large strides. His head is lifted high up. There are some whiskers trailing on the ground, but the whiskers aren't making much contact. Let me show that again. You see the whiskers are barely moving back and forth at all, but what we can do is we can show that the whiskers are pushed forward. So relative to their normal state, they're all pushed ahead, they're protracted, and it appears to be that what he's doing is he's using them to try and anticipate collisions. So rather than using them for exploration, he's using them for what we might call look ahead. So this is the quantification of these results, and it comes out very nicely that if you plot what you might call the set point, the average angle of the whiskers, and you plot that against the velocity at which he's running, the faster he's running, the further forward the whiskers are pushed. And this is his running velocity plotted against the amplitude of whisking, and the faster he's running, the the smaller the amplitude of the whisker motion. So he's going from doing these big sweeps at slow speeds to doing these little sweeps with the whiskers pushed forward when he's running quickly. And this is really independent of whereabouts in training he is. So these colored dots are showing you the different stages of training. Green is when he's naive, but actually the relationship seems to be with velocity rather than with familiarity. So there doesn't seem to be an effect, particularly for familiarity. If he's not very familiar with the environment, but he's still running fast, then he does small sweeps and he pushes the whiskers forward. So this is some data that we're collecting. We haven't analyzed yet, but we are looking now at when you place objects on the floor, which interrupt his progression. He doesn't like to stand on edges like the circles in this piece of Perspex. And so when he contacts this, you can see him exploring with his whiskers and using the whiskers, we think, to decide where to place his feet. So this is work that's in progress. So I want to now talk about the...
Question.
Yeah.
Have you done any natural habitat sort of experiments? Because these floors are smooth, the walls are smooth.
Yeah.
They're not really three-dimensional.
Well, this is about as natural as it gets in the world of whisking research, because it's very difficult to film the whisker movements unless you can light them properly. So we have underfloor lighting here. We have an experiment where we brought the whiskers closer together, to the walls closer together to what would be a typical burrow width of a natural rat habitat, and we don't think that makes much difference to this result. So if the walls are closer together, as they might be in a burrow, we think we will still get this effect. But
Another quick question.
Yeah.
Is there any lateralization of the whisker sense? In other words, so they purposely use the left side or the right side for exploring?
Yeah, I think, I don't think anyone's analyzed that specifically, but I think there's some impression that there may be some lateralization, but I wouldn't like to.
And that could be a population bias, not an individual
I think there certainly needs to be looked at. I don't think there's a paper that describes it, but I wouldn't rule it out.
Those two curves that you showed were not the obverse of each other, those two correlations. Is there any meaning to that?
Well, you mean is there an dependence between these two things?
Yeah, it looked as though each of them had breakpoints.
I think there is, if you are pushing the set point forward, there's a point at which, if you want to maintain set point this far ahead, then you can't do big amplitude sweeps. So I think these things aren't very independent. So we want to do some more quantification of this, because it might be possible to say something about the relationship between the velocity with which he's running and how much time he has to anticipate things which are happening in the world. So obviously, if you're running faster and you want to give yourself the same amount of time to anticipate something, then you need to have information from further ahead in the world. So that's something that we're going to work on with these data. So on to whiskered robots. So this is the guy we're trying to copy, and this is part of the robot brain. And these are a succession of robots that we've built. There's another one not shown here. We started out in 2003 building this guy, Whiskerbot. I won't show you any more of him. His whiskers, his head doesn't move, and his whiskers move back and forth. He had a maximum of 3 whiskers per side. Then we built this guy, Scratchbot. We realized after building Whiskerbot that really his head not being able to move was a real limitation in terms of trying to understand how the rat uses its whiskers. So Scratchbot has a much more animal-like neck and many degrees of freedom, therefore, for positioning the whiskers. He also had independent actuation of 3 columns on each side of 3 whiskers each. And then we built this modular sensor, which we could put in different configurations. And here you can see it on the end of a robot arm. On this cone, in this case, had 8 rows of sensors. In this case, 2 of these rows are populated with 4 whiskers each. So here we can do individual control of each whisker if we want to. So we wanted to use this system to try and understand the architecture.
Question.
Yeah.
what are the temporal frequencies that you use in your robots?
You mean the
whisking, like?
Okay, well, we generally whisk at about 4 hertz in the robots rather than 8 hertz in rats. I think that there are some scaling factors here. So if you look at larger rodents, they tend to whisk slower than smaller ones.
Is that an engineering limitation, or you just choose that frequency?
I think we could go faster if you wanted to. It depends what actuators we use. We've used a variety of different ones. So we use BioWire. The maximum we could go, I'll show you in a minute, that we got up to 4 hertz with that. I think we could go faster. But generally, we want to be able to measure what's going on in the whisker tips, so it's better if they're moving more slowly. So this is a picture of the rat brain, and this is the somatosensory area, very large area of cortex, much bigger somatosensory area than visual or auditory. And this is a rough outline of the architecture, the control architecture for the vibrissal system, which many people study, but a lot of the time they study this part of the architecture here. So from the vibrissae, there's an ascending pathway via the trigeminal complex and the thalamus to the sensory cortex, this area in the brain here. And you'll find that maybe 80% of the papers on the vibrissal system are talking about what's going on in the sensory cortex, particularly in the S1 area of cortex. And Partha mentioned the barrels, so this is a blow-up, I think this is from Matthew Diamond, of the somatosensory area. And within the somatosensory area, you find an area which is specifically for the vibrissal system, and for each of the macrovibrissae, but also for the microvibrissae, you find a patch of cells which can be identified by staining that primarily respond or have the strongest response to one whisker. So if you deflect this whisker here, then you know that you can record in a specific bunch of cells in barrel cortex and measure something about the brain's response to that deflection. So this is what makes this system so attractive to neuroscientists, because you can have a very controlled deflection of the whisker in an anesthetized animal, say, and you can exactly know the parameters with which the whiskers moved, and then you can look in a precise spot in the brain and measure the response of the cortex to that stimulation. And this is why it's proved such an attractive system in which to try and understand not just somatosensory processing, but cortical processing generally. But that's just one of the loops. So another important loop is one that goes through the brainstem. So it's possible for inputs from the vibrissae to go via the brainstem to the facial nucleus, which then controls the movement of the whiskers. So there's a low-level loop which allows feedback from tactile sensing to influence whisker movement. Then, of course, there's a midbrain loop, and this goes via the trigeminal complex to the superior colliculus, and that also can influence whisker movement, and I'll talk more about that later. The cerebellar nuclei can influence this loop. In fact, they can influence many of these loops.
Do you leave your telephone in your pocket or close to the transmitter of the microphone?
Okay. And I've put the basal ganglia in here as well. It's been mentioned a few times, and we've done a lot of work to model the basal ganglia. And the basal ganglia is having an effect on all of these loops. It has outputs that can affect the brainstem, the midbrain, and the cortical loops in this system.
With my newfound anatomical knowledge,
Yeah.
a prominent projection from the motor cortex to the sensory cortex, so those arrows are not just going in one direction.
Yeah, exactly. I mean, we're not showing anything like all of the arrows here. This is a simplification of a diagram that Ehud Akisar and David Kleinfeld put together. And I'm really just using this to illustrate the loops and not all of the connections.
One would think sensory to motor, but actually there's a lot coming back from motor to
Yeah,
sensory.
and actually I think it's probably not that useful to think of this as sensory and this as motor, because this has consequences for the motor system, and this I think has consequences for sensory processing. So we should think of this as sensory motor loops, and these labels can be misleading.
So is there a translation of that the system has ray afference signaling, a corollary discharge?
It's actually interesting because there isn't a spindle cell in the intrinsic muscles. So you can't actually record the position of the muscles directly. but there are signals ascending from the follicles, or certainly the skin around the follicles, that correlate with the position of the whisker. And so it does seem to be recording the position of the system and to be using that information, relaying it back to cortex. So the system does know something about where its whiskers are in space. And that's important for us, I think.
So it's derived from an open loop sort of motor command rather than a proprioceptive...
Well, it's probably getting the open loop information as well, but it's certainly recording from sensors around the whisker, something about the position, how much the skin is stretched perhaps, and then that's correlating with where the whisker is in its sweep. So I want to very quickly give you a flavor of the robot models. There's no time to go into details here. So a key thing to do obviously was to model what's happening in the vibrissal, in the whisker itself and in the follicle that it sits in, very complicated object, and the trigeminal ganglion, the earliest phases of processing. So this was our first artificial whisker. We took it to SFN. We were very proud of it. And this is it moving. This is around 4 Hertz. It's driven by this piece of bio-wire, which is kind of an artificial muscle. So we take the signal when the whisker taps against an object. We take the signal, we feed it through an electro-mechanical model of the follicle. This is a blow-up of the follicle from Ibarra. There's a whole hundreds of mechanosensory elements in here. There's lots of other tissues, soft and hard. So we model this as a spring damper system, and then we generated spike trains based on this, and we showed that we can get simulated spike trains from our artificial whisker that fitted some of the data from the biological labs. So we think we have a clear idea of the kind of processing that's going on at the level of the follicle.
So I didn't catch, what is the transducer there actually? Is it a force transducer? That's,
So in this one, we had a strain gauge at the base of the whisker.
oh, okay.
But in more recent models, we're using Hall effect sensors, magnetic ones. So I'll just go through quickly through the next loop. So this is the midbrain loop by the superior colliculus. And what we have implemented is that the colliculus controls orienting, and I'll come back to this a bit later if I've got time. But the basal ganglia provides a mechanism to decide whether or not you should orient when you make a contact. Because, of course, every time you touch an object, you have the option of exploring it or ignoring it. And if the rat is in an alley and the object's there that it's seen before, it won't always turn and explore. It's only a novel object which will elicit an orient. And we have worked a lot on basal ganglia models on the assumption that the basal ganglia is there to one of the components of the system that decides whether you should select a particular action. So the basal ganglia can inhibit an orient action if it has other priorities. We also model the cerebellum, and in this case we use the cerebellar model to remove sensory signals that are due to self-movement. I'll briefly tell you about that. Let me just show you Scratchbot. This is orienting. So this is Martin Pearson with a ruler that he's placing in the whisker field, and you can see Scratchbot orienting, putting its microvibrissae close to the point of the whisker contact. So this was Scratchbot about 4 or 5 years ago doing this, and you can see lots of movements of the neck. So the positioning of the whiskers is not just about controlling the vibrissal muscles, it's also about controlling the head and neck muscles. But everything is driven by the point in space. So he selects a point in space to target, and then the control of the muscles goes from there. So to select a point in space, given a vibrissal contact, you need to do 2 things. You need to find the coincidence between which whisker made the contact, and you also need to know whereabouts in space your whisker was when it made that contact, because the whisker is moving back and forth doing this sweep. I mentioned before that there are these, the cell population in the primary afferents which appear to signal position within the sweep. Those are called the theta cells by Ikurakisar. And so if you have a contact on one of your cells that indicate contact with an object, so there's a population that responds to contact, a population that appears to encode angle or some correlate of angle. If you have a a coincidence between contact on a particular whisker, so label line code, and some knowledge about the angle of the whisker when it made that contact, and you have some coincidence detector neurons, and David Kleinfeld has suggested there might be some of these in S1 Cortex. I think we can also hypothesize there might be some lower down in the brain too. Then those coincidence detectors could tell you whereabouts in space that contact happened. So in our model of the colliculus, we have, this is Scratchbot making contacts with an object here, and we are representing in 3D space the position of where the contacts are happening. And these are what are guiding orienting, or could be used to guide orienting. We'll come back to this later. Quickly mention what the cerebellum is doing. So Paul Dean and John Porrell in our department have spent a long time developing a theory of the cerebellum as an adaptive filter. And one of the roles of the adaptive filter could be to predict sensory signals that are due to your own movement. And if you look, for instance, at the electric fish, these animals have a very large cerebellar-like structure, and there's strong evidence now that what that cerebellar-like structure is doing is it's generating a cancellation signal, so that when the fish moves, and the fish is generating an electric field, which it uses to catch prey. If the fish moves, that of course affects its electric field, but you can use the motor signal to anticipate what effect that will have on the electric field. And the cerebellar-like structure in the fish appears to do that. It takes the motor signal, which is generating the movement of the fish, of the tail or the fin, and it is using that to say what would be the consequence in terms of the effect on my electric field, and then it can remove that signal from the electric field signal to find out what's left, which might be due to some prey animal. And a very similar idea is happening here, that we say, well, if the whisker is sweeping through the air like this, then just sweeping my whisker through air could generate some force at the base of the whisker, which I'm detecting with my strain gauge. And I don't want to respond to a signal that is due just to the movement of my whisker through space. Without this system, Scratchbot might detect some activity in the base of a whisker and generate an orient to something when there's nothing there. We called it a ghost orient. So we put in this adaptive filter model of the cerebellum. We can predict what sensory signals could be due to the movement of the whisker itself, and then we can cancel that, and we get very good noise reduction by using this model. So there's
exactly the noise reduction, because the output pathways from the cerebellum and essentially give you an excitatory response from the nuclei. So how are you then adding or subtracting that from your signal?
Well, yeah, so
the silent versions.
So I think the model is that the output from the cerebellum is predicting the sensory signal due to self-movement, and then the cancellation is happening.
Let's say this is more done in a more abstract fashion in this model.
Yeah. Yeah. So I don't think we're proposing this happens in the cerebellum, but that the cerebellum learns to predict the sensory signal due to the movement, and then somewhere in the brain you can cancel out those aspects if that's what you want to do.
You get this vector response from your deep nucleus, which essentially is excitatory.
Right. Yeah.
Right. Now, and this is your control signal, right?
Yeah.
Your signal.
For sure. So yeah. So we're taking that predicted noise signal and we're cancelling it with the noisy whisker signal somewhere in the brain and getting a cleaned up whisker signal. Certainly that's what we're doing in the robot. So, and then finally, just to mention what the basal ganglia is doing. So we previously did a lot of work on the basal ganglia with Peter Redgrave and Kevin Gurney. This is going back to the mid-90s when we started this work on action selection by the basal ganglia. And I'll refer back to this later. In this case, Scratchbot has a small group of behaviors. One of them is orienting, but another one is just generally exploring. In order to orient to a contact, he needs to decide that I'm going to stop exploring now and I'm going to investigate this point in space that I've identified something interesting might be at. And so we worked before on various models of the basal ganglia, suggesting how they might be involved in making sensible decisions. Nathan Lepora, who's here, is Nathan here today?
He's on the beach.
Yeah, he's on his way. He's here next week. He's been working a lot recently with Kevin Gurney to develop our models of the basal ganglia and show how they may implement forms of optimal decision making. So, how am I doing for time?
Great.
Good. Okay.
But look, a question then. Since we have so much time. I mean, earlier Partha already asked you about, look, you have to sort of unidirectionality in the scheme, right? But that might be questionable.
Yeah.
But now also you make a clear separation.
There's no unidirectionality, so maybe
wait, but
slide
I
by
want to ask
slide.
you on something else.
Okay.
So it's apart from the directionality, that's done.
Okay.
So now we're going to go for the next one, which is this clear layer distinction. Because
Yeah.
in some sense, basal ganglia cannot do very much if it doesn't receive inputs from the cortex.
Yeah.
Right. So what allows you, in some sense, to draw a strict boundary there? Is that reasonable?
Okay. So that's a good question,
Thank you.
and I think that the answer is this: that there are two things going on in brain architecture. And we wrote a paper about this in 1999. There's at least two things. One of them is a layered scheme with the brainstem, midbrain,
Right.
and forebrain loops, certainly in the sensorimotor systems. Another thing that's going on is what Penfield called the centrencephalic system, which he identified with the basal ganglia and the reticular formation. And I think this links back to the previous talk, that this central system, basal ganglia and reticular formation, goes across these layers. And it's an integrative system that samples data from each of the layers and is therefore able to help prioritize your behavior. Because you don't want to risk your brainstem loop and your cortical loop deciding to do different things and then getting some competition of motor commands at the motor output side. So you want to do something consistent across your whole brain. We think the basal ganglia is involved in doing that. So the primary thing that basal ganglia does is inhibit things. It's inhibitory both of the cortex and of the brainstem. In order to select a voluntary action, certainly, the basal ganglia has to remove that inhibition. And it does that by selecting particular pathways from which the tonic inhibition is going to be removed, and then that system can be released to execute a behavior. So that's our general model that the basal ganglia is doing this.
There is inhibition both at the cortical and the brainstem level, and one of them can be selectively removed?
I would hypothesize that's the case. I wouldn't necessarily say I've got evidence for that. There's certainly inhibition at the cortical and the brainstem level, and the basal ganglia is configured to remove the inhibition from particular target systems, and one type of target system is muscle systems. That might remove inhibition at the cortical level and the brainstem system at the same time. Now, whether the brainstem loops can directly compete with the cortical loops is an interesting question. I would think they might. Peter Redgrave and John McAfee wrote a nice paper about the role of the basal ganglia and its interaction with brainstem systems.
Coupled, right? Like the cerebellum.
Exactly.
It has dense projections to frontal cortical areas.
Yeah. So I think most of the time these things are working in concert.
Right.
Okay, so in this rapid tour of different loops in the brain, I wanted to say something about S1 Cortex. This is data from Carl Peterson's lab, which I think is really beautiful data. It's actually collected by Farazu, published in 2007. I think it was in Neuron. And what they did was took the skull off a mouse here, and then I think it was an awake animal, deflected the C2 whisker. And this then is the C2 barrel in the mouse. And this blue spot here is to indicate motor cortex. And this maybe comes back to your question about the reciprocal relationship between cortex, because what you'll see when we play the video is how the activity in the C2 whisker barrel spreads across the brain. Let me play the video. So what you see is that the stimulus occurs here, and then this is the activity in the S1 barrel here, the red. You can see it spreading out, in fact spreading out across most of the brain. So this is really a change in membrane potential. It's not firing, but it's giving us an idea about the general activity across cortex. And you can see that activity in motor cortex emerging with very little time lag compared to the activity in S1 cortex.
That's the response to the whisker being deflected in one direction, is it?
That's right. So this is a simple whisker deflection, and then you get this response. And you get also, so this is the contralateral cortex, and on the ipsilateral side you also get some response eventually.
But totally, just for clarification,
Yeah.
the motor cortex, so the behavioral response of the animal, there is an overt behavioral response to this?
I think it's head fixed, so no.
Is this calcium imaging?
Yes, voltage diode imaging.
It is ending up in the middle. what regional is that it is going into?
In here, you mean?
Yeah, there's like a spot. It's like a specific. It starts out in S1, goes to M1, and then seems to go to another little spot.
Yeah.
Yeah.
Well, I'm not sure what's happening. It's going over here really as well. So, but one way of thinking about it is ripples spreading out from a central point, and this is what we've been doing when we've been modeling it.
It almost looks like there's two waves that collide in the middle.
Yes.
It's interesting.
So what you're saying happens certainly at a smaller scale here. So this is a result from Schmeggi, 2000. So they looked at if you deflect two whiskers at the same time that go to adjacent barrels, or you deflect them and you vary the time lag between whisker A being deflected, whisker B being deflected. And they looked at cells that were between the two barrels or directly in one of the barrels. And they found a facilitation effect, particularly at the border between the two whisker barrels. If you deflect two whiskers at the same time, then the cells which are directly at the center between the two barrels have the strong facilitation effect, much stronger than to one whisker alone. So this cell here would be one of these central cells, and if the two whiskers are simultaneously deflected, it gets a strong response, which is super additive compared to the individual response. And so one way to think about both of these results is that the activity can be thought of ripples spreading out across cortex. So these are imagining the center of three whisker barrels and an object deflecting the whiskers moving through the whisker field and triggering these ripples, and where the ripples contact, you would get these strong responses in barrel cortex. And another way to think about this is that you're taking temporal information about how the object moved through the whisker field, and of course also about the shape of the object that was moving through the field, and you're converting it into spatial information in terms of positions across the barrel surface that encode something about the relationship. So if these two whiskers were deflected at the same time, you'd expect to get the strongest response in the middle. But if, as here, this one is deflected first and then that one later, then the strongest response would be much closer to the center of the second barrel.
So, as you know, right, we have shown that if you take the amplitude time course of the bliss population response,
yeah,
this will give you a very powerful encoding scheme, right?
Yeah.
It goes beyond this intersection approach.
Well, I'll show you in a minute what we've been doing with this. So we can build a very simple model of this based on this geometry of the barrel cortex, and assuming sort of realistic values for the transmission times. Also in our model we have inhibitory cells as well as the excitatory ones to make it work. And we can fit the Schmeggi et al. Data, that's the original data up there, and this is our simulation data. This is Stuart Wilson's work published in PLOS Bio earlier this year, and we get a nice fit to this result. So we think there's evidence for this spatial this temporal to spatial code conversion in the barrel cortex. So something else that Stuart's been doing for quite a long time with Jim Bednar in Edinburgh is to develop self-organizing models of what might be going on in S1. So these are using Hebbian learning. This one was to try and explain the finding of directional sensitivity and a directional map in S1 Cortex. So here we're using Hebbian learning, and this is just showing objects moving through the whisker field. This is a 5x5 grid of simulated barrels. And this is Hebbian learning going on when objects move at different velocities and directions through the whisker field. And this is, over time, these are sort of pinwheel maps which were originally described by Anderson and Moore as being found in the barrels of rats. And this is actually quite a controversial result, because although they found these pinwheels, so this is actually the Anderson-Moore result down here. So what you see here is within each barrel a preferred direction of whisker deflection for the neurons in the barrel, and nearby cells in the barrel have a similar preferred direction. And like the pinwheel maps you get in cortex, if you move around in the barrel, then nearby regions would have similar preferred directions. So Anderson and Moore showed this, I think, in rats that are a few weeks old. Various studies failed to replicate this, but a recent one has shown that if animals are of the right age and have had the right experience, they do have these pinwheel maps. So the pinwheel maps are down to learning. They're not something that emerge automatically. And these are the pinwheel maps that we've developed with our model. And Stuart has been looking more recently about the more complex responses you can get, particularly layer 5 of S1 Cortex. This is work by Jacob from Dan Schulz's lab in Paris. And in layer 5, you get cells whose response to an object moving through the whole whisker field is different from its response to if you tweak an individual whisker. So these cells still have a preferred whisker to which they respond most strongly, and they have a preferred direction for movement of that whisker. But their preferred response when all of the whiskers are moved is for a different direction of movement than from single cells. So these are showing response properties to global changes or global movement of whiskers, which are quite subtle and could indicate things like shape. And so Stuart has recently been looking at self-organizing models that can encode things like shape, direction, and speed of objects moving through the whisker field.
So, but Tony, these neurons then give you an object encoding in their rate response? Or how do you see that?
I think so. Well,
like a grandmother cell, basically.
Well, they're showing a preferred direction for movement across the array and deflection of multiple whiskers. So,
but then you said something about object detection, right? The recognition.
Well, so if you want to distinguish, for instance, a object that's moving through the field which has a straight edge from one that's got a curved edge, then these kinds of responses could give you that kind of discrimination, and that's what Stuart's been looking at. So he's been generating artificial data with this whisker array here, using different straight edges and curved edges, convex and concave, and showing that you can develop different maps in a simulated layer of cortical neurons which encode for these properties. So, so I want to leave the cortex there and go back to active sensing. So I was showing you before this evidence that the animal controls the whiskers so as to make contacts of in a particular way. So it does things like moving the whiskers closer together to investigate a surface, changing the symmetry of the whisker movements in a way that appears to create more contacts with the surface, and controlling the timing of the contact so that when it makes a contact with the surface, it starts to retract quite quickly. So we've done various experiments with robots to try and establish that controlling the whiskers in this way will have an effect on what the sensory system experiences. This is an experiment with a robot called Shrewbot that I'll show you more of later. And Shrewbot was a redesign of Scratchbot. You see it has these modular whiskers, so we can individually control each whisker now on Shrewbot. And the whole shape of the nose cone was changed to make it morphologically more similar to what we see in the animal. And the point of this picture, which was from a 2011 paper we published, is to show that if you control the whiskers in a way that simulates some aspects of this active control, then you can generate more contacts on the whiskers than if you just have a stereotyped movement of the whiskers. This was an earlier experiment with Scratchbot, and the goal here was to show that if we so if this is just a movement of the whiskers and they all move together at the same speed, then what happens, of course, is that the front whiskers contact first and bend hard against the object. The more caudal whiskers contact late on and hardly touch at all. But if in response to that initial contact, the whiskers are then controlled so there's inhibition on the more rostral whisker and excitation on the caudal whisker, then obviously this one will bend less, and this one will make more contact and bend a little more. So you can go from this initial pattern of activity, very strong response on the front whisker, virtually no response on the back whisker. But with this feedback scheme, you can then generate more uniform contacts across all of the whiskers. And our hypothesis is that the rat is aiming for uniform contacts, because then within the range of contacts that it expects to have, it can have more accuracy. You can represent that range better.
It's like a gain control.
Yeah, exactly. Yeah. So we do have some evidence, and Nathan has been working on this. This is the cone sensor, and this is investigating different textures. And we have some evidence that this active touch control and this effect of lightly touching surfaces can help us make some sensory discriminations. That's kind of work in progress.
But you are then trading off your bandwidth, right, in some sense?
Yeah, I mean, so I don't think it always helps, and we're investigating this further. So we're actually going for shorter sample times, but we're trying to get more uniform types of samples. So sometimes it works, and sometimes it maybe doesn't help. So we're trying to analyze the circumstances under which it's useful to do.
So if you trim one of these whiskers, it would change its
yeah, it would actuate longer in order to make a contact. So I want to switch gears slightly and on the next section of the talk about evolution. So I'm sorry that Nick has had to leave, but I'm wanting to kind of follow on in the path that he was establishing us on yesterday in thinking about the evolution of the brain. And I want to try and persuade you that the vibrissal system is a really interesting system when we're thinking about mammalian brain evolution. And I want to give you some insights into what the vibrissal system might tell us about the evolution of mammals. So this is a fossil of an ancestral mammal. This one, I think it's called Eomaya, and it probably lived maybe 125 million years ago. And this was from a paper by G et al. In Nature, 2002. This is from a paper, I think, by Liu et al. It's more recent. And this is really just showing you geological time here from about 200 million years ago. So if you remember yesterday, Nick was talking about stuff that happened in the lower Cambrian. So 500 million years ago, so that would be well below the floor here on this chart. So 200 million years ago, still a long time ago, but it's actually within the Jurassic, so the time of dinosaurs. And animals that were the precursors to modern mammals were around, but much more reptilian than mammalian. And then what happened between 160 and 170 million years ago, it's proposed, is that the first mammals really took off. So you see here actually the monotremes, which are things like the duck-billed platypus and the echidna, breaking off from the rest of the placental mammals, actually back here, sort of 180, 190 million years ago. And then the common ancestor to all placental mammals, so these are animals that give birth to live young, appearing around here 160 million years ago. And this recent fossil find I'll show you in a minute, Jeremiah, dated to about 160 million years ago, is possibly an example of what these early placental mammals might have been like. And then the placental mammals break into two groups. One is the marsupials, so you see up here a kangaroo, and they break probably at this point or close to it, and then the rest of the placental mammals here, which includes ourselves. So this supergroup is called the Therian mammals, and Jeremiah is an example of what an early Therian mammal might have been like. Let me just show you this picture. This is Jeremiah. So this is the fossil find by Liu et al., published last year. This is a reconstruction of its skeleton, and this is an artist's rendering of what this animal might look like. And Nick yesterday was talking about this idea of living fossils, of animals that can tell us a lot about what some of these stem creatures might have been like. And this is an animal called Monodelphis domestica. And it's a marsupial opossum. It has a lot of primitive features which suggests that this animal, Jeremiah, and Monodelphis domestica may have interesting similarities. So if we compare these two animals, we may learn, if we look at this animal, we may learn something interesting about these stem mammals from the Jurassic era. So what we've done, and this was what we published last year, was a comparison of the vibrissal system in this opossum, Monodelphis domestica, with two rodents. So we took mice and rats, and we compared them with the opossum. So as the previous diagram showed, these animals split a long time ago, 160 million years possibly. Monotreme split a bit before that. And so if these two animals share common features, one hypothesis is that the ancestral mammal also had those features. And the feature we're looking for is active control of the vibrissal system. So let me show you some of the data. So this is, what we did was I think is the picture here? Yes, we took this arena and our high-speed camera to a lab in Trieste in Italy, where they have a colony of opossums. And so we recorded these opossums running around in this arena. So it's very similar to what we were doing with our rats in our own lab. So here you see rats. This is a rat exploring in our own laboratory. You see the whiskers moving back and forth. This is automatically tracked. And what we're calculating here is the average position of the whiskers over time. And you see it follows this nice wave. And we can fit this wave with a Fourier time series, and we can show what the frequency of the oscillation is, and we can calculate the amplitude. This one is a mouse, and you can see that we can also fit oscillations here. They look a bit more variable, and I'll talk about that in a minute. It looks like it's oscillating faster, but I'll explain that the story is actually more complex than it appears here. People have often published a result that mice whisk at 20 hertz, whereas rats whisk at 10 hertz, but actually I'm going to try and persuade you that that's not quite the case. And then this is a monodelphis, and this is a typical trace, so you see very smooth, stereotyped whisker movements here, as this is the opossum. So this is very slowed down, so you can see some whisker motion there as he's coming up to explore this object.
Tony, I don't understand the drift in these two graphs.
So this would be the set point of the whiskers. You mean the vertical drift?
Yeah, that's just because of head movements, or?
Well, there may be some measurement error, but also he may be whisking further forward here than he is there. So there he's whisking far back, and then he moves and starts whisking
right, okay.
further ahead of himself. So, and this is work that Mitch did. What he did was actually take a two-component Fourier series to try and fit these curves. And so one of the things he did was to take out the set point so that you can remove this vertical drift that you can see. And then, so that would be this line here that's not oscillating, would be the set point. And then have two Fourier components with different frequencies and amplitudes, and see if you could get a good fit that could explain a lot of the variance. And so this is a fit for the rat, the mouse, and the opossum. And I just want to focus in on what we found here. So this is the frequency, or the base frequency of the stronger component. And this is in the rat up here, so you see a frequency of around 8 hertz. And this is mouse, and it's faster, but it's not that much faster. It's around 11 to 12 hertz is the base frequency that we found in mouse. So this was contradicting other people's results who were saying 20 hertz. I'll explain why in a minute. And this is a possum. We found a possum was really quite variable, but the average was similar to rat, about 8 hertz. What I suspect actually is that the opossum's frequency is a bit more locked to locomotion speed, although it's something we'd have to investigate to further demonstrate that. Or maybe not locomotion speed, but other things like sniffing. It could be because of the nature of the pattern generators.
Right.
So, and there's an interesting story to talk about we can discuss later if people are interested.
So one thing that I think Kleinfeld showed is that the range of frequencies, a given bout has a given frequency, and a different bout is the same story here.
Well, it depends very much on what the rat is doing. So David Kleinfeld showed that sometimes when a rat is investigating an object, they appear to whisk at a higher frequency than the base frequency.
Well, he showed that you saw like a fixed frequency bout and another fixed frequency bout.
So I think the frequency probably varies around 8 hertz for rats most of the time, but sometimes you get what he calls foveal whisking, which is where they're exploring an object and they appear to be whisking much faster.
The sniffing frequency goes up too, doesn't it, generally when they're doing olfactory sampling?
So what people have been looking at recently is the correlation between sniffing and whisking, and showing, well, there's two papers that rather contradict each other, but I think the general conclusion is that they are linked. And Martin Deschênes has recently got some quite good evidence suggesting that the pattern generator for whisking is driven by the pattern generator for sniffing, so that the pattern generators are actually coupled. Though I think that the cells in the reticular formation that are the pattern generators for whisking are not the same group of cells for sniffing. So there could be some possibility for a looser coupling. So they may not be directly correlated under all situations. So what I wanted to do was to quickly show you this result about the second component. So this again is rat, mouse, and possum. And what it's showing here is the amplitude of the second component and the phase shift relative to the first component. Now, the first thing to note is that what we found for all of these animals, that the second component, when making a best fit to these traces, was always at double the base frequency. So the appropriate second component was a harmonic of the base frequency. So essentially, you have a whisking base frequency, then you have a second harmonic, which appears to modulate it. And so this is an example of rat whisking at the top. It's really just the base frequency. There's very little second harmonic. Here you have one with some second harmonic, changing it. Here you have quite a strong second harmonic, generating these sort of smaller whisks in between the larger peaks. Some people have described this as kind of double pump. And Wineski in 1983, who studied whisking in hamsters, described this phenomenon that he saw of them doing big whisk and then a small whisk. And that is actually a lot like what's happening here. This is from a mouse. You can see a big pump and then a smaller pump, big pump, smaller pump. And this is the combination of the second harmonic with the first harmonic. So the base frequency is around 11 hertz, but then there's a second harmonic of that, which is offset, which is generating this other peak. So when you look at the mouse whisking, and I wouldn't say that the rats don't ever have a strong second harmonic. It's possible we're just not eliciting it in this task.
It's very depending upon the nature of the process. The third harmonic might be more important than the second, right? I mean, for example, if you have a saturation or something, then you get the odd harmonics coming into the picture and the even harmonics don't exist. So I mean, it really depends on the nature of the beast, I guess. Look at the third harmonic, the next higher harmonic?
I think we'd have to look back at the methods and see whether the search extended up to as far as the third harmonic would be. I think we probably did look in the range of the third harmonic, and in that case we can't have found anything, otherwise we would have reported it.
But the explanation that he gave for big whisk and the small whisk would be consistent with the second harmonic.
Sure.
Yeah.
What's the contribution of, let's say, the passive deflections of the whiskers to this?
Well, I mean, this is...
also give you some harmonics.
Yeah, so this is, in this study, they were always whisking over the floor, so there was some influence of contacts. And this is some reason why it'd be useful to look at other data. So many people collect data in animals that are head fixed and are whisking in air. There's a big problem with that, in that animals don't spontaneously whisk when they're head fixed. So you have to induce them to whisk by putting something interesting in front of their nose.
Right.
And I think certainly if you put an object in the whisker field of a head-fixed animal, it's not the same as a freely moving animal encountering an object. The head-fixed animal will just take this as a threat, and it will do sort of retraction, and it will try and escape from it, whereas the freely moving animal obviously will think that's interesting and want to explore.
Just a very quick question about this synchrony between the whisking and the sniffing. Does the inhalation coincide with the whisker going forward? And the exhalation with the whisker going back? Just out of interest. I mean, I know it's got nothing to do with what you're saying.
I don't know. I'm sure that data will be in Martin Descend's paper, though. So I think, I don't know. I can imagine that you'd want to sniff at the same time you're making the contact, and therefore inhalation would correspond with the maximum protraction. So, and I think so. This is an interesting story about a possible difference between mice and rats, two species that are supposedly quite closely related, and we study them in the lab. We tend to think of them, certainly in terms of the whisker system, as being very similar, but the mice have expressed this much stronger second harmonic, certainly in our data. And if other people find this and find a weaker second harmonic in rats, then it's an important species difference we should take into account when thinking about the system.
Amplitude phase plot.
Yeah.
There are also the mice seem to be in a very different ballpark than your other two species.
Yeah, because the amplitude is much greater. So this is really showing you that.
It looks so extremely different. This is
exactly.
strange.
Yeah, yeah. Well, I think mice are very different animals. It may be because they're smaller size. So we might want to look at other small rodents, because there may be something about the physical size of your whisker that it's easier with these smaller, lighter whiskers to generate faster.
There could have been something in the experimental environment, maybe. Some artifact of your experiment. Chemical cue, I don't know. Because it looks really so extremely different.
Well, I mean, that's always possible, but it was certainly the same setup for the mice and the rats that we used. So, I mean,
could you think of engineering reasons as to why it would be useful to have this double peak? One thing that I could see is it's a shorter contact period. That would be useful.
I mean, I think certainly you can imagine reasons why it's better to sample faster. So, if you could double your sample rate, you could get, in principle, a lot more information quite quickly.
For texture discrimination, I think you want to have a high frequency sampling.
Yeah. And then maybe there's some advantage to be able to do a big push and a small push. I don't know. These are things that we haven't looked at yet, but they're good questions. So I wanted to show you the evidence for the active control of the whiskers. All right, okay.
Interesting. To decompose this now in, let's say, the detection of different features of objects, because you have something like a texture, which might need a high frequency sampling, and then you might have something like a solidity, for which you might need a more slower but with more force.
Yeah.
So it might be an interesting way to decompose now these components.
Yeah, no, it could be interesting.
Idea, right?
Yeah, definitely. So, but I think also sometimes you see this in mouse, and sometimes it doesn't do that. So we can probably get at this behaviorally. If we can look at the circumstances under which this happens.
Two different pattern
in a more controlled way.
generators?
Huh?
You'd say they're two different pattern generators?
Well, I would say that since this is always at the second harmonic, it could just be the first pattern generator and then something else modulating it, which would be consistent perhaps with the base frequency being tied closely to sniffing, and then some other reticular formation component generating the modulating signal that can double the frequency and shift the phase. So it would be nice to see if that could come out of Martin Deschênes' work.
Is there temporal structure of this? That means that when the animal encounters an object, you see predominantly certain frequencies, and only in the second whisk you see the other frequencies dominating. Is there any regularity like this?
Well, we haven't investigated that far yet. So other people have found evidence of these kind of double pumps, but nobody's really systematically investigated when they happen. What might be interesting to speculate also is that maybe the opossum, because the opossum shows very little evidence of a strong second harmonic. So possibly there's a whisking pattern generator evolution going on in modern rodents. So maybe in the opossums it's more directly coupled to sniffing. That'd be interesting to look at. So just to show you the active control results. So this was, remember, head turning asymmetry, where when the animal is turning, the whiskers go ahead and explore the space that you're moving into. And this is original data from Towle and Hartman, and this is data from the rat, the mouse, and also the possum, showing that when they're turning their heads, they do tend to asymmetrically move their whiskers in the same way in all of these species. This is the contact-induced asymmetry. So this is what happens when there's a contact on one side, and the whiskers on that side move a lot less, and on the other side they move a lot more. And this is our way of plotting this. So this is the ipsilateral side. Red means less movement. This is the contralateral side. Blue means more than average movement. So in the rat, you see the strong effect, the ipsilateral side, less protraction, the contralateral side, more. We see it also in mouse, a bit noisier perhaps, but very clearly also in the opossum you see this effect of contact-induced asymmetry. Now this is all the more interesting because the opossum lacks corpus callosum. So like all marsupials, there's no direct contact between the two halves of cortex, so the signals that are mediating this must be crossing over lower down in the brain.
Could there be behavioral coupling in any way?
And how could it be behavioral?
Movements, whatever.
Well, this is all in either under infrared or with blind animals.
But I mean, if there's a contact further to the right, there might be a compensatory movement, and then there's an additional, like a reduction of sampling on the left, or It's a behavioral pattern that gives you this bias, as opposed to an internal public, like behavioral feedback. Is it feasible?
I don't think so, but I'd have to think about that a bit more. What we have shown in the previous study, I don't have the data here, is that this isn't an effect of just interacting with the environment. You can actually see it if you measure in the intrinsic muscles with EEG, that the EEG is stronger on this side and weak on that side. So it's definitely that he's driving these whiskers harder.
So even if it's moving in free space without making any mechanical contact with anything, the whiskers leave the head when the head turns, is what you're saying.
No, sorry, that was the head turning asymmetry. That was the first result. So this was originally, Mitra Hartmann showed this in animals that were whisking in space. So she was getting them to explore over the edge of a platform. So there was no contact with anything. In our experiment, the animals were exploring across a floor. But even so, when they were turning their heads, they were showing this head turning asymmetry. I mean, maybe not as strongly. So the slope of this graph is weaker, perhaps, but there's still a strong effect. So contact-induced asymmetry is where there's contact with an object on, and if you look at it when there's contact on one side here, then these whiskers barely move at all. They're pushed back against the cheek, and these ones really push round. And when you watch the videos, you can imagine that the animal knows there's something there and it's trying to reach it with its whiskers. And so these are other plots showing you this change in the whisker field. You can see the whiskers here pulled back, these ones pushed forward. So what we're arguing for is that this first mammal, so if opossums and rats and mice diverged 160 million years ago, and they all do this active control of the vibrissal, then a hypothesis is that an ancestral mammal like Jeremiah, also had active vibrissal sensing. So that's certainly possible given those data and the similarities between these different animals. An alternative hypothesis, of course, is it's convergent. So they all had whiskers and they separately evolved active control over the intervening period. So evidence that it was not convergent but a common ancestor would be if there was similar mechanism underlying active whisking in all these species. So what we did was to look at the vibrissal musculature in the opossum. So this is the rat up here. So this is data from Sebastian Herderloo in Ikuraxar's lab. So these are the follicles. These are microvibrissae here. That's the rat. These are the follicles after you've done a slice and you've stained to find the muscles. And these brown lines here are the sling muscles. And the characteristic architecture in rodents is that these sling muscles join together pairs of follicles. So each follicle has a sling around it, and that sling goes to the next follicle and is attached to the base of the next follicle. So it's a rather unusual architecture, if you like. And then that happens along the row. So each pair of whiskers along the row is connected by this sling muscle. So we wanted to see if this was replicated in the opossum. So this is now the opossum snout. You can see the follicles. It actually has no microvibrissae, interestingly. It has a gland on the tip of its nose which it uses to put scent on things. So I don't know if scenting means it can't also have microvibrissae. But it has prominent macrovibrissae, and it also has these very nice sling muscles joining the whiskers within the row. If you actually look at the whiskers on the face, it looks less organized than on the rat. It looks like a more primitive system. The moustachial pad looks less developed on this animal, but the musculature inside the pad seems to be very similar. So there's sling muscles within each row. It's common between rodents and opossums. And that to me suggests that the common ancestor, it's unlikely that this same architecture would evolve twice. So the common ancestor would have had these sling muscles, and therefore most likely would have had whisker movement and active control of whisker movement.
Two questions, Tony. For clarification, functionally, if you would remove these sling muscles, it would mean that you lose the coupling between your vibrissa.
So,
right? So, what's the functional relevance of these muscles?
So, what happens when the animal whisks is that there are a bunch of extrinsic muscles, and these are the intrinsic muscles.
Yeah.
So, the first part of the whisk is extrinsic muscles, sort of anchored in the snout here, which pull the whiskers forward, or they pull the moustachial pad.
Yeah. They pull the whole pad.
So and that does the first part of the protraction. And then these whiskers, these muscles kick in and they do the second phase of the protraction.
Okay.
And then there are some other muscles back here in the cheek which pull everything back, and also a certain amount of elasticity in the system that pulls it back. So that's the retraction is mainly by the extrinsic muscles. So these muscles are really just doing the phase 2 of a protraction movement.
Okay. And then what's the earliest example of, let's say, a vertebrate who would have whiskers that are not actively moving? That's just implanted in the snout but not under control. Where does that end?
So whiskers are hair, and hair is new in mammals. So the first hairs were probably tactile hairs, because it's difficult to conceive of how you could evolve hair for thermoregulation until you have hair for something else, because hairs don't have a useful role in conserving heat until there's lots of them. So people who have thought about this have proposed that tactile hair was the first hair. The most common place to find tactile hair is on the snout. So every mammal species, bar humans, at some point has some tactile hair on the snout. Yeah. Yeah. So I think this would be they're not hairs, so that would be evolutionary convergence.
What are they using it for? Do you know?
Well, sometimes it's tactile, sometimes it's chemosensory, but I agree, there's lots of species that are using these kinds of sensors.
Yeah. Your original question is to see whether their common ancestor has active whisking.
Yeah.
In some sense, what you're saying now could be like, well, as soon as you put a whisker in your snout, it's going to be active.
Well,
so is there any example of an animal that has whiskers sticking up its snout that are not actively whisking?
So there are lots of animals that don't whisk, or don't whisk very much, that have whiskers. Things like dogs and cats, for instance. But they do have muscles.
They do have muscles, in
yeah.
theory.
Yeah. So, but they don't whisk. So.
So you think it's a loss of function?
So I take that. Well, I don't know. I don't know if we can say that the first mammals, as soon as they evolved hair, were whisking. So, but I think as soon as they evolved tactile hair, and it was probably, the tactile hairs were probably at the front of the animal, because that's where you get useful information. Remember that the current hypothesis about the evolution of mammals is that they were largely nocturnal, because reptiles dominated the day. So these were small animals, they were nocturnal, they were developing thermoregulation so that they could be more active at night. That meant they could be move around a lot faster, but they were also metabolically much more expensive, and so they had to eat a lot more. So they have to be more efficient predators. So you're a predator moving around at night, probably in quite a complex environment, so quite possibly living in bushes or whatever there was in the Jurassic, sort of off the ground a lot of the time, climbing, trying to find prey. And what you're trying to do then is use information from vision, which is going to be pretty sparse and noisy, combine that with information from audition, but also from this tactile sense. So, if you're on this limb of a fern, you want to see if you can jump to another one, you can use your whiskers to reach across the gap. So I think this might be one of the things that's pushing the evolution of tactile sensing.
When you see some answer, I would sort of say maybe your original question is not exactly rightly phrased then, because if the conclusion is like, well, as soon as you put the whiskers in there, they will be active. And your question is whether the common ancestor has active whisking or passive whisking might not be the right question to pose. Or I misunderstood it.
Well, I don't think you can be sure that as soon as you have whiskers, they would be active.
Right, exactly.
So I think this data suggests that early on in mammalian evolution, we not only had whiskers, but we also had active whiskers. So there are some. There's very little in terms of fossils of pre-mammalian reptiles, but the assumption is that they may well have had whiskers of some kind before we get to this boundary with the Therian mammals. Okay, so I think we've pretty much covered this. So the other thing to say about Monodelphis domestica, so it's a great model animal. So Nick was talking yesterday about why don't we study more model animals, why we restricted to, in this case, rodents, rats and mice, because the few people that do study Monodelphis find it a fantastic model. It's born very immature, it's born after just 2 weeks gestation, and it's this tiny little grub that then crawls up the mother and clings onto it for months. So if you want to study development, it's a great animal to study development in. It also takes 6 months to mature, so it takes a rat about 3 weeks to mature, and so you get maturation over a much slower timetable. So if you want to look at development in slow motion, it's a good animal for that. It doesn't have hardly any corticospinal neurons. So it's an animal in which the cortex is not able to directly influence the control of movement anywhere except perhaps in the vibrissal, because Frost et al. Demonstrated that if you do microstimulation across the cortex, if you microstimulate somatosensory cortex, you can get movement of the whiskers.
Out of curiosity, that's pretty amazing, right? That few corticospinal movements.
Yeah.
So how is the cortex affecting behavior then in that system?
By basal ganglia.
Okay.
Largely.
Of course.
Wait, so it is, how is it? So basal ganglia is disinhibiting cortex, and then...
Well, basal ganglia will be selecting the different bits of the motor system that it wants to enable. There's a nice study looking at comparison of forepaw movements between monodelphis and rats, and showing that the forepaw movement is a lot less sophisticated in monodelphis. So if you want to get, sophisticated control of digits, that's when the motor cortex appears to be useful. So this is an animal in which we can start to think about why the motor cortex, or why the cortex evolved a motor aspect, because this animal has very little motor function, and but what motor function it does have in cortex appears to affect the vibrissal system.
You're saying it's largely a sensory organ.
Well, it's not doing much in terms of motor, is what I'm saying. But if we want to think about the evolution of cortical motor function, then let's look at this animal, because we can look at vibrissal control of movement, and we can look at how S1 here is regulating the control of the vibrissal. And this is why I think it's one of the reasons I think it's inappropriate to call this somatosensory, because it really has a motor output, even in this animal that's directly affecting movement. This is just to show sort of a comparison of different species, and this is the opossum here. So you see, actually has these guard hairs at the back, and the whiskers are quite far back on quite a pointy snout. This is a shrew, even pointier snout, and the whiskers quite far back. So the ancestral condition probably was for the whiskers to be further back and sort of radiate outwards. But you can see in these modern rodents, these are all examples of modern rodents that we've filmed, and they tend to have a much blunter snout, and the whiskers are much further forward. So you can see that they've evolved the specialization for whisker sensing. The whiskers have come forward relative to the tip of the snout, and they're also denser.
So the old idea that the whisker length is roughly proportional to the width of the body, so it tells the animal whether it can go through a gap,
yeah.
And that fatter animals have longer whiskers. That's all no longer true?
I think that might be one of the functions of the whiskers. So see, this animal and many animals like it, the opossum, has these guard hairs which are back on the cheek. They don't seem to have quite the same musculature, but that's definitely detecting gap size, whereas these are doing something else. So interestingly, rats have lost these guard hairs, or at least they don't have them to anything like the same extent. In terms of whisker length, I just want to show you this video here. So we've been looking at the dormouse, and the dormouse is interesting because it's very distantly related to rats and mice. It's in a different branch of the rodent family, and these guys actually have the longest whiskers of just about anything, and their environment that they exist in is in bushes. So to move along a twig like this is what the dormouse would do most of its time. You can see enormously long whiskers which are being used here to help it navigate along this twig. And you can see here a lot of asynchrony and asymmetry in the whisker motion as he's exploring around. So these animals are protected in the UK, but we can study them using high-speed video like this. And going back to what I said about how the whiskers might be important for moving on irregular surfaces, what we'd like to do, if we can get funding, is explore how this animal could use the whiskers for climbing. Okay, so I'm on to the last section. We still have some time, yeah?
Yeah, you're doing great.
Yeah. Okay, so I want to come back to this question of attention now, and how we can use the whisker system to think about attentional control in mammals and how we can model this in robots. So the observations that I described to you of active control of the vibrissal system, we think we can summarize most of them with a term that we call minimal impingement, maximal contact. So minimal impingement is this idea that when the whiskers touch against an object, they don't bend hard against the object. They make a light touch. It's minimal impingement because if you imagine that the whisker was moving through the surface, that would, if it made a hard touch, it would move a long way through the surface. So you're trying to minimize the amount that the whisker could move through a surface or bend against the surface. And at the same time, you want to maximize the contact. So you want to have as many whiskers as possible make contact with the surface while making the contacts with the surface quite light and gentle. And I think this generalizes to human touch. If you imagine you're exploring with your fingers, you're not pressing hard. You're finding the surface, and then you're touching with really quite a controlled amount of pressure as you move around and make different contacts.
You might also want to assess the elasticity of the surface, I suppose.
Yeah, so if you're interested in compression, then you might press harder. So it depends on the task. And that's one of the things that makes this active sensing, is that depending on what information you want, you'll use different strategies. If you're interested in texture, you'd run your finger along. If you're interested in shape, you might palpate different positions. If you're interested in compression, then you squeeze. So, and there's some evidence that rats have different strategies in that way as well if you vary the task. So going back to this video, the hypothesis then is that vibrissal behavior can reveal details of the rat's spatial attention. So eye movements can reveal the point in a picture that you're interested in at a specific time. And that can tell us something about perhaps what you're thinking or what you're interested in. The foveation of the rat, where it puts the tip of its nose, can tell us a lot about what the rat is interested in. But I think the whisker system can also tell us something about the area in the world that the rat's interested in. So it's not just a point, the way that eye movements give you a point. It's a zone. And the whiskers can be configured so that they explore a large zone around the head or a small zone that they can foveate around. So it's a nice system in that we get more than one parameter of attention here. It's not just the point in space that's interesting, it's the zone and the size of that zone that we can get from this system. So what's driving the control of attention in the rat vibrissal system? So this is the most recent thing that we're thinking about. How is attention controlled? More generally, how is the behavior of the system controlled? So in the earlier talk today, we were thinking about behaviors and how you choose different behaviors. An idea that goes back to ethologists, this is from Behrens, is that you could have a decomposition of control into different behaviors. So this is a very famous idea. Behrens used the nesting behavior of a herring gull to illustrate it, so that there'd be different components of behavior like settling, building a nest, locomotion, trimming and bathing, and there might be different control systems that would implement these different aspects of behavior, and that they would inhibit each other so that you would only do one behavior at a time. So this kind of behavioral decomposition of control. And if you look in parts of the brainstem, like the periaqueductal gray, you can see some evidence of this kind of behavioral decomposition of control. You can stimulate in parts of the rat brainstem and get some components of behavior in this way. So, and this is actually what we did in Scratchbot. So when we wanted to build Scratchbot and get it to orient to stimuli, we decomposed it into 3 different behaviors. It had explore, so it would just be moving around, investigating objects and the floor with its whiskers, or it could find something interesting and orient to it, or when it got bored of orienting, it could back off and go somewhere else. And these 3 behaviors would inhibit each other and be selected via the basal ganglia. And this is now Shrewbot implementing pretty much that control system. So exploring in space, finding something interesting, orienting, investigating it, then backing off and moving away. If we copy the ethologists and draw an ethogram, and we can actually generate this ethogram from what we know about the control system, so we can generate it automatically. Then initially it does an explore. Let me just run the video again. Initially it does explore behavior for a few seconds. Then there's a switch to this orienting behavior. Then there's a switch to the back off. And then he goes back to exploring. So this is one way for decomposing the control of behavior. And it generates appropriate sequences of what look like motivated action.
That's very nice, and it suggests that it looks very natural.
Yes.
And it suggests that people may be fooled by this. Have you tried to take people who are naive to
yeah, well, people that interact with this robot do treat it like an animal. Because it's,
I mean, there's a big business in the toy industry, I imagine.
Yeah.
Or maybe sorry. I don't know.
you could fund your research.
well, this is what we try to do. So, but by going to European conferences mainly and trying to impress the evaluators. So this is one way of decomposing behavior. But there's another way we could think about decomposing the selection of behavior, which I want to illustrate by first going back to visual attention. So there are many models now which suggest that the way we generate eye movements is that we look at the visual scene and then we generate a saliency map. So you decide what are the hotspots in this image, and then you select a particular hotspot, maybe this one because it's the strongest, and you foveate there first. And then after a while, looking at that, you say, okay, well, this one's strong over there, I'll go and look at that one, and so on. And so you move from hotspot to hotspot. And there's something called inhibition of return that stops you going back repeatedly to this first one. But after a while of exploring the image, that will wear off, and you will go back to this early hotspot. So there's now a lot of these saliency map models which are being used in robotics. This is another scene, different set of hotspots. And of course, it's not just the bottom-up information about edges and contrast that's driving this saliency map. There's a lot of top-down information about, I'm interested in people because I'm a person, and so on. So how about applying the saliency map idea to the control of attention in our robot? So this is a very generalized version of the loops model that I showed you earlier. We have a selection mechanism. You could imagine the basal ganglia. We have a model of allocentric memory. I haven't had time to describe to you, but we are building models of the hippocampus and the role that it might play in encoding tactile spatial information. And we have a way of controlling the movement of the body. So we have, and we may have a selection mechanism that basically uses a saliency map. So we have a representation somewhere in the brain of 2D, 3D space, and we decide what's important, where should I focus my attention, and we use that to drive behavior. So here we're doing that for Shrubot, and so now what's happening is not that Shrubot is moving through a different set of behaviors and selecting each one in turn. It's being driven by a foveal point of attention, which is shifting around according to some criteria, which I'll show you in a minute. So the behavior looks a bit different here because it's running actually a different control algorithm for whisking. But it's generally pretty much indistinguishable, the sequence of actions that Shrubot chooses compared to what we were doing with the previous system, the behavior selection system. And this tells us what we already know, that you can't just from looking at the behavior of an animal infer the decomposition of control within the brain of that animal. So I want to go back to whisker motion. I'll just show you this guy. It's a Coendo from Washington Museum. And although he's eating at the moment, which is rather confusing his whisker motions, he can really move his whiskers as well. So it's not just guys with short whiskers that move them. So what we looked at for the generation of these active sensing strategies, first of all, was could we do it with a reflex loop? Could we think the brainstem is using feedback from the trigeminal complex to modulate the facial nucleus and just drive, for instance, the front whiskers faster than the back ones? And we're running out of time?
Wow, so
yeah, not that long to go. Okay, so we looked at reflex loops. You could have a inhibitory loop that reduces movement of the front whisker, excitation to the back whiskers. You could generate some of these behaviors, rapid cessation of protraction, head turning asymmetry, with these kind of reflex loop. What you can't really do is generate look ahead, because for look ahead, there isn't a direct sensory stimulus which is telling you that when you're running down a corridor, you should be paying more attention to what's in front of you. We also found when we tried to control our robots that actually to tune some of these active sensing behaviors by implementing them as reflexes was very difficult. This is partly because there are delays in the system, there's noise in the system, the position of objects in space relative to the whiskers changes depending on how you approach them. It's very difficult to actually tune on the physical platform a reflex mechanism so that it always gives you an appropriate light touch. So although we could do it to some extent, we didn't really like it. It wasn't very clean, we weren't getting clean signals. So what we have been looking at more recently is a stronger assumption about what the rat is doing and the possibility that the rat is not just using reflexes. It's representing the environment. It's representing, much in the same way we think of salience maps for human attention. It has a salience map of where are the hotspots in my environment that I want to control my whisking behavior. If I have a hotspot and it's this size and this shape, then I could control my whiskers specifically so that the whisker tips will land inside that zone. So that's our hypothesis. What we wanted to do was to build a model that could implement this idea of there being an attended region and modulating the movement of the whiskers on an individual whisker basis so that they would end in this region. So it's a high-level model. So we have information about contact going in here. We could have some other motivational signals driving it too. We have the whiskers. We have information about head position and head movement, which can drive head turning asymmetry. The contacts can drive the contact-induced asymmetry. To actually implement it, we also include some inhibition of return, because like humans, you don't want to always be going back to the same spot in the environment. That would look unnatural and be boring. We have a foveal zone, we have an oscillator generating whisking, and we have the physical model to implement this on, which is one of our robots. This is more detailed, breaking it down. So what we have actually is an inhibitory zone. So we inhibit anything that's inside the head of the animal or the robot. We inhibit anything that's inside what we think is a solid object in the environment. And then we have an excitatory zone that we think is a region where we'd like to explore with our whiskers. And so we combine these two. This is the zone, the salience zone that we want to explore, and we use that to drive the movements of the head of the robot or the simulation, and also to configure the movement of the whiskers, so the asymmetry and so on.
So if you present the robot with a surface that's pivoting, moving in front of it,
yeah.
pivoting, could you build an anticipation of the next instant, for example?
Well, let me I'll show you the video in a minute, which is exactly that experiment. So these are just simulation results. So this is with a 2D simulation that Ben Mitchison built. So this was the biological data here from rats, from our lab, and from Towle and Hartman. We can simulate it quite nicely with our model. So these slides are all the same, showing you the model of contact-induced asymmetry. It looks a lot like the biology. The model of whisker spread, bringing the whiskers closer together. That's similar to the biology. But this is more fun to look at the video. This is the experiment you just talked about. So this is a new whisking robot, sometimes called Goosebot. I don't know if that's the best name for it. We have another degree of freedom here I've not talked about, which is we actually move with these linear actuators the rows back and forth, and this is to try and replicate some of the movement capability of the mystachal pad. And so this is being controlled by a version of the system I've just described to you. And the idea is that we generate as many contacts as possible on as many whiskers as possible while doing it with a light touch. And as you can see in this experiment, the board is being moved around all the time. So the thing is being reconfigured in real time to know, so it changes its estimate of where objects are in space with that movement. So I'll show you this video as well, which this is Ben Mitchison, the developer of this system, who for some crazy reason decided to take his glasses off to do this experiment. So I think what he's demonstrating here is that it really is quite a light touch, because this is a very powerful robot arm that could give you a really good punch on the nose. So the next phase of this experiment will be perhaps to get Paul and Anna and various people to put their heads in front of this, and then we can show that we can do face discrimination. When it's Paul's turn, we will give him a punch on the nose.
Is the whiskers of the same material as the shoe ball?
So yeah, these are the same modular whiskers now in a different configuration.
They're very soft, so I touched the shoe ball.
They're not that soft actually, I don't know. they're reasonably soft, yeah. So you can, with Shrubot, people naturally come up and they let it move the whiskers against their legs and hands. So this is just a little bit braver than that. So I'm just about to wrap up. So I wanted to just say, okay, if there's a saliency map, where is it in the brain? The obvious place to look for it might be in the superior colliculus. So in the eye movement literature, one of the dominant theories is what might be called the gaze displacement hypothesis. That the deep layers of the colliculus provide a gaze displacement command that tells you how to move your eyes and your head to the points in space which are interesting to you. So what we are advocating is what you might call the whisker displacement hypothesis, that somewhere in these deeper layers of the colliculus there is a whisker displacement command that tells you how to move your head and your whiskers in order to explore this attentional zone that you've identified. So this is data from Draeger and Hubel showing that in the colliculus there are lots of whisker responsive cells, and actually they seem to be broadly tuned, and they seem to be tuned to different whiskers. But one explanation of that is that these cells are tuned to points in space, and so as your whisker moves through space on its arc, it will enter the receptive fields of different cells. So this is why the C2 whisker could turn up in different cells as it moves across its arc. It will move across this map that was in the colliculus. So there's nice data showing also that these cells get very fast signals from the input, so that you can get a signal within about 5 milliseconds here. This is of the whiskers being deflected, so there's the possibility that the colliculus could drive some very fast reflexive behaviors. And finally, this is data from Hamilton and Keller showing that colliculus can change the amplitude of whisker movements, which is just exactly what we want. If we want to change how much they protract by, is we want to be able to change how far forward the whiskers are pushed. And so motor cortex seems to be more involved with whisk frequency, and colliculus seems to be able to change the amplitude, perhaps also the set point, the position of the whiskers in its arc.
How does the signal get from the colliculus to the cortex in
the model? The main thing we're interested in is that colliculus talks directly to the facial nucleus. The facial nucleus generates the whisker movements, but the cortex talks obviously into the colliculus. So this second peak here in the colliculus is the signal from S1 Cortex. So S1 Cortex modulates what's happening in the colliculus.
I guess when you're talking about attention, it's not like cortical activities being modulated. You're thinking of something in the colliculus that then, because normally when people talk about attention, they kind of have cortex in mind.
So what I would say about cortex is that it's directly modulating this map in colliculus. So the attentional effects could be coming down from cortex, for instance, from S1. So the map that generates the whisker commands would be in the colliculus, but the other components that regulate attention, for instance, motivation, could be coming from elsewhere in the brain. The basal ganglia talks into the colliculus and is able to inhibit or disinhibit it as well. So I think I'm finished then. So this is just that first video now slowed down. So you can see the whisker movements perhaps a bit more clearly. You can see that rats and mice and many other animals have these really quite exquisite and I think quite precise movement of the whiskers. They can vary depending on what the task is. Here doing long sweeps, exploring over the edge of the windowsill. Here doing asymmetry, exploring with contact on one side, not the other. Here you can see him gently touching around this object, trying to find out something about its properties. We now, I think, after about 10 years or so of work in this lab and others like Mitra Hartmann, we now know a lot about the parameters that he's controlling in order to regulate these movements, and about the environmental influences and feedbacks which are regulating these movements. And as a result of all the massive effort on the neuroscience side, we also know an enormous amount about the sensorimotor loops, which are controlling this behavior. And I think we're now in a good position to use this system to look at attention and perhaps to answer some of these questions about attention, which are not so easily addressed in primate models. And in addition to studying animals, we think we can also make some contribution by studying robots. This is Schubot on a day when he was being a bit less cooperative and a bit less interested. So we think that we can use these synthetic systems to embody our hypotheses about how this system works. At the very least, we can show that it does what it says on the tin, that the system can demonstrate the behaviors. Quite often, we think it also generates new hypotheses, which we can then take back to the biology. Thank you very much.
All right, any questions for Tony? All right, Alex.
In the robot studies, what were the signals or the information that you extracted from the strain gauge sensor?
So,
Can you repeat the question, Frank?
so the question was what information we get from the strain gauge sensors on the whisker. So we've actually had several different kinds of transducer. So initially we're using strain gauges, but we found they were a bit unreliable. When we used strain gauges, we would put 2 strain gauges on the whisker so that we could get bending in 2 directions. With the Hall effect, we also generally use 2D, so we get bending in X and Y. With the Hall effect sensor, you can also get Z, so you can get movement of the whisker into the follicle, if you like. And there's evidence that rats do use that quite a lot. So if you imagine you're pressing the tip of your whisker against an object, then you create some pushing force which can be transmitted down the shaft into the follicle. So that could be useful for finding out when you've touched something. So these are just the raw data sort of bending. We can then either do one of 2 things. We can turn it into a spiking signal if we want to investigate or model early processing. But more typically, we just take the raw bending measures and use that for our model. So we bypass the neural signaling.
Signal or just the average over time?
No, it's sampled. I'm not sure what the granularity is.
If you move the whisker, you get a temporal profile.
Yeah, exactly. Yeah.
The question is, do you use the full information or do you somehow compress it into a single value?
No, we use the full profile, but what I can't tell you is what the rate that it's operating on. But it's fast. I think it's, but I'd need to check. It's probably in the papers.
Sure.
So if you were to test a real mouse with surfaces of varying degrees of compliance, I mean, I was wondering, so the signal for it that tells it when to retract the whisker after it's made contact would be a force signal, I presume, that it's sensing? So if you had a more compliant object, it would push it even further before it retracted, right? Yeah,
well that's an interesting hypothesis. I think Mitra Hartman's talked about looking into that. I'm not sure if she has any results yet. So far, every lab I know is just using hard objects.
Okay. Can I ask one last question?
Yeah.
Sure. Shut up. You mentioned that in the beginning, towards the early part of the lecture, you were saying that when a mouse runs faster, it tends to place its whiskers further forward. So some sort of maybe assessing time to contact with some object or something. The faster you. So if you trim a mouse's whiskers, would it run slower?
It might adapt to run slower. I mean, so this is a good question. We might even have some data. So Peter Redgrave in our lab did clip the whiskers off some animals, but we didn't run them in our arena. Maybe it's something we should do. I imagine in the dark they would move more slowly, but we need to demonstrate that.
Armin.
Just in general, what are the capacity of rats to discriminate objects or surfaces with the whiskers? Is there any data that would show what is a little bit resolution of this sensing capacity?
So people have looked mainly at texture discrimination, and the claim is that they have similar ability to discriminate texture as the human fingertip. So they can tell the difference between a completely smooth surface and a very finely milled surface that you or I would only just be able to tell.
Really? Because I think ours is pretty crappy. You know? I think with these whiskers you will have more.
Well, it might be better, but it's at least as good. So the the experiment I'm thinking of, they took these tubes and they were completely smooth, and then they milled them for different textures, but they found a version of the tube which hadn't been deliberately milled but was so finely altered that they could hardly tell was actually discriminable by the rats. So it's pretty good at texture. There's not much investigation of shape discrimination, I think, because shape discrimination is really a multi-whisker problem, and people tend to study what's happening with a single whisker and then the correlates in cortex. So I think that's where the interesting research in the future will be people like Dan Schultz is able to, has an array that can move the whiskers, many whiskers at the same time, and therefore present different patterns to the whiskers and look at cortical activation. So with that kind of apparatus, we can look at object shape and things like this.
So Tony, another issue. So what's the visual acuity of these rats in the area where their whiskers are active? Is there any overlap?
Yeah, there's quite a lot of overlap, and in fact it's interesting that the study on the colliculus suggests that the visual system does set up the topographic map in the colliculus, and then the whiskers are overlaid on that, which is odd because the whisker system matures before the visual system.
Right, but you would need that if you want to use your whisker, the same whisker, to make statements about different points in space. This is how you were interpreting the collicular map, right? You were saying that the responses there relate to this proximal space.
Yeah, yeah.
And these whiskers are moving through that space and then contributing information to, if you want,
yeah,
to neurons to colliculus, and that would imply that their scaffold has to be visually anchored.
No, not necessarily. So they just have to have, need to know their position in space relative to the head. Which,
well, I was saying whether vision could do that. That was my question.
But vision could contribute certainly to that. So we also are working on the possibility that the cerebellum is really involved in calibrating this map. So that's work in progress.
Oh.
So that so one of the things that happens a lot, of course, is the whiskers break and they grow out. And so you're continually having to recalibrate your idea of where your whisker tips are. And the cerebellum could be really critical for this, which is why there are many large areas of cerebellum in the rat which are devoted to whiskers, and nobody really knows what most of them are doing.
So you have to explain it over lunch then?
Well, I can try, but Paul Dean would be the guy, because he's leading that
Right,
investigation.
All right, so Tony Prescott, thank you very much.